So now we have another industry talk from BioLab, I guess. Digital PCR. Yeah, so new kind of PCR you can buy the machine.
Hi, very good afternoon to everybody. Hope I'm audible. Yeah. So, hi, this is Dr. Abhijit. I represent BioLab Laboratories and I'm here today to give a brief talk about a newer technology in the market called as a droplet digital PCR. So, just in brief, what I will be doing is I will be discussing about the technology, its applications, and what are the advantages that you can have as compared to the conventional technologies. So, I would just request if anybody has any questions, concerns at any point, you can stop me. I'll try my best to address it. So, just in brief, I'm sure all of us know this. This is just how the life cycle of a PCR has evolved. All of us know what we talk here of a first generation PCR. We put a thermal cycling reaction, we run it on a gel. If we see a band, the DNA is present. If no, it's absent, right? Then came the second generation of PCRs, which is called as a quantitative PCR, or more commonly called as real-time PCRs, okay? Where it's always assumed that we get to a quantitative value of your nucleic acid, but a very important thing to take a note here is the second generation of a PCR, or real-time PCR we're talking of, is always a relative quantification. You need to have certain standards, you need to have certain housekeeping genes, controls, only then you have a deduced value coming out of your data and you come to a value which is telling you a quantity of nucleic acid. So a lot of processes involved, a lot of calculations, a lot of discrepancies. Then we come to the third generation of PCRs here, which we call it as a digital PCR, or more specifically because I represent the brand, it is called as a droplet digital PCR, which the major advantage is it gives you an absolute value of your nucleic acids. So the data that you get is in copies of your nucleic acid per microliter of your sample. You don't need to run any standards, no need of housekeeping genes, no need of calculations. It's a fully automated system that will give you a precise quantity of your nucleic acid in the sample. Okay. Moving ahead, the major step of differentiation of how a digital PCR is different than a real-time PCR is the principle of partitioning. Okay. So in this case, what we are doing is we have a same reaction mixture that we make for a real-time PCR. That 20 microliter reaction is now split into 20,000 compartments, okay? And now there are 20,000 parallel PCRs which are happening, which are working together, and each of those 20,000 droplets are counted individually. So now you have a combination of 20,000 readouts from one 20 microliter reaction, okay? Just as an example, if I would have to show you this, now if you are walking or if you are standing on a pond and somebody asks you a very basic question of tell us how many are yellow fishes, how many are red, how many are orange. It's practically impossible, right? But if we now remove those and put them in individual bowls, and then if you are asked to count, it's easy, right? So this is exactly what we are doing here. We have a compartment, we have individual compartments which are now amplifying your nucleic acid, and then you detect and calculate each one of them individually. Okay? The major advantages that you get with the ddPCR as compared to any other conventional quantitative PCR technology, first we are talking of a precision, okay? So we have umpteen publications where we have shown that even a single copy of your nucleic acid, let it be a DNA or RNA, is precisely quantified, is precisely given as a data point, okay? Coming to the second point which I already mentioned, this technology is an absolute quantification method. You don't need standards, you don't need reference genes, no calculations involved, no interpreted data. It is a direct quantity of your nucleic acid which is presented to you. Okay. Third point coming to is the multiplexing. This gives you a flexibility to use a dye-based chemistry also or a probe-based chemistry also, and in either of those cases you can do multiplexings. Okay. So there is no need to worry that you have to run at one gene one time. You can have multiple genes in the same tube and you get precise quantity of each one of them. Okay. Next advantage, sensitivity. I already mentioned it can go up to a single copy detection very precisely. Again, just to add on to a point here, the advantage here is even if the quality of your nucleic acid is bad, if you have degraded DNA, if you have like liquid biopsy, if you have nucleic acid from your paraffin blobs, right? even those type of nucleic acids can be precisely quantified using this technology. And the last and the most important point is tolerance to inhibitors. Even if your sample has tons of inhibitors, it is not going to work on any other technology, but this will still precisely quantify your nucleic acid and give you a precise data. Okay? So these are the major advantages of having this technology. I'll just quickly explain the workflow, what happens here. So in the first step, what we simply do is, we have a normal reaction mixture which we make for a real-time PCR. We have your primers, probes, nucleic acid, supermix, and that is now split. This is exactly how it works. So there is an oil, there is your nucleic acid reaction mix, it makes droplets. It makes droplets of exactly same size, same volume, right? And now these droplets we put back in a PCR plate which goes in a thermal cycle. And now that's where thermal cycling happens. And then we have another small instrument which looks like this, which is called as a droplet reader. So my friend who gave a talk before this from ED, right? So all of us understand how a flow cytometer works, correct? So this reader is exactly like a flow cytometer, only difference being a flow cytometer counts cells. The same principle here, this counts each droplet, right? So there are multiple lasers sitting inside the reader. Each droplet is going to pass through in a collinear manner, and there is a precise detection that happens, and that's how you get a precise quantity of nucleic acid. And the data that you get is something like this here. So a scatter plot that you get on a real-time piece or a flow cytometer, a similar scatter plot is given here as well. So this what you see here, this is the data from per well, okay? So each of this dot represents one droplet. So you're not missing out to anything. The droplets which do not have a nucleic acid will have no fluorescence. They would sit at the bottom right here, right? And the ones who have fluorescence are here. So that's how they are scattered, that's how they are plotted. And a similar thing in a 2D, you see it here. So anybody who understands flow cytometer, you have a FAM positive, you have a X positive, you have double positive and negative. So similar data is there, and the software then interprets further, converts the digital signals into a quantitative data which is given to you precisely. Okay. I would skip this because of time constraints. Yeah. This is just again a 2 months back old data still. So today we have got close to 6,000 final publications using this technology. Okay, across the globe, across different applications. In India, we have close to 100 installations now with this technology. A lot of people getting advantage. So those of you who are working on any kind of a nucleic acid amplification process, I would urge you to take this into consideration. And this is how the life cycle has gone. So Bio-Rad has been a proprietary manufacturer of this technology. It's been 10 years, it's been just a decade now when we have started this, and these are the different product lines that we have crossed over a decade. And with a lot of pride I can say we have expertise, we have support, we have global knowledge base which can help you understand this better. Okay? And these are just the applications broadly in which this technology has been used, starting from viral titers to agriculture to food biotechnologies to water detection, somebody working on gene expression, mutation detection, all of it. Basically any kind of nucleic acid work you want to do, this can answer your concerns. Okay? What I now do quickly is a couple of slides where I would just show you a few snapshots from few publications, okay, where this technology is helping. So this was one of the publications which is from Harvard, if I'm not wrong, where a precise 1 copy to 1 lakh copies of your viral were loaded in the sample, and that's how a precise detection of that was done with this technology. Right? Again, coming to next study, this is again from the publications where this was carried on across 5 different countries in probably 5 different parts of the globe, right? Similar sample was sent, different machines, different time points, and this is how a precise data was represented at the end of it. This was close to some 5,000 odd samples across different ports. Okay? One more very important, I would say, feather in the hat for us, the National Measurement Institute in Australia, which does validations for most of the controls that we get for different instruments, right? So this is an instrument which is used for validations of those known controls, right? So this is one of the data which they had published across some 5 lakh samples, and you see in the second graph less than 2% CV. This is how reproducible, this is how precise this technology is. Okay? Again, a very challenging application for those of you who work on copy number variations. If you want to differentiate between 1 copies, 2 copies, it's still easy, right? But when you go to differentiating 5 versus 6 copies or vice versa, right? That's when the problem comes. And this is the ddPCR, again one of the publications where you see the copy number differences. And the last part is where the same was done on a well-optimized real-time PCR technology. You see the variations there, and you see the precision with the ddPCR. Again, this was from Stanford, so I mean, you can be rest assured of what was published is what was standard. Okay. Again, one of the very critical applications when someone goes to single-point mutations, right? Below 1%, the real-time PCR is never going to detect your mutation frequency. And this is again from one of the publications where right from 0.001% mutations all the way till 1% by multiple users have been verified, and this is the precision with which this technology is detected and quantified in nucleic acid. Last point I would focusing upon, again, which becomes a concern for a lot of us who are using a real-time PCR, is inhibitors. Right? Our reactions don't work and we don't know why it is. Okay? So again, this is from one of the publications where a lot of inhibitors were purposefully added to the sample just to check how rigid this technology is. And as you see, you have media, you have HEK cell lysate, you have DMSO, EDTA, and still the technology has precisely quantified without any variations and given you the quantity of nucleic acid. And just the last part of it, where I was talking about the multiplexing, certain publications where with a dye-based chemistry also, people have done multiplexing up to 4-plex assays in a single tube, and these are a few publications. And this is from a probe-based chemistry, where using a Tachman chemistry or any other FAM heads with TAMRA or any other quenchers. This is how people have published up to 8-plex assays in a single well with this technology. So yeah, that's how it is represented for the single positive, double positive, and the negatives. And just as a last thing, if someone is using a real-time PCR, I mean, it's not that you have to do something great. So this is a tech note and a publication that explains you how a well-optimized real-time can directly be replicated for this technology. You don't have to do any efforts. Just with the added advantage, like you don't need standards, you don't need references, inhibitors, and the most important part here is your LOD and LOQ is always the same. Your limit of detection and limit of quantification is always the same. So somebody who use a real-time PCR, your LOD will always be higher, because any fluorescence coming out of your machine, it will get captured, but that cannot be quantified. But here, whatever fluorescence is coming in, whatever nucleic acid quantity is there, the machine will precisely detect as well as quantify it. So that becomes a major advantage. And I would like to stop with that. If there are any questions, I will be more than happy to answer.
Thank you, Abhijit. Anybody have any questions?
Yes, ma'am, please.
As you showed, the digital PCR has so many advantages over the real-time PCR, but what's about the cost effectiveness?
Ma'am, yeah, very good question, ma'am. I will tell you point blank, this becomes cheaper as compared to even your real-time PCR. In CPT, what we call as cost per test, because in a real-time PCR, practically to get one meaningful data point, you need to have your 3 wells minimum as your control gene, 3 wells for your sample, your negatives, positives, so practically close to 10 to 12 wells for getting one meaningful data, which is still a relative quantification. Here, your one data point is your quantified data precisely.
I think it will be less cheaper.
It is cheaper as compared to real-time PCR.
We have to analyze so many samples, but if
Yes.
I have very few samples, I have to do the digital PCR. In that case, I think it's not cost effective.
No, ma'am, it is still cheaper. It is still cheaper because, I mean, I did not get time to explain you the entire workflow. You can practically run one sample to 96 samples at the same go at the same time. And this is, ma'am, there is no difference. Your real-time PCR reaction, what you make, is still going to remain the same. The only part that is different here is your partitioning in the first step, where you're making droplets. That's the only additional point. Other than that, your reaction mixture and everything is the same that you do for real-time PCR. So there is practically, I would say, a 10% increase in your price if you're talking of a single well. But when multiple wells are used there as replicates, here's a single data point from a single well. So all of that is nullified and this becomes a cheaper technology compared to real-time PCR in terms of running cost.
Experiment is cheaper and the machine is costly.
Yes, rightly said, sir.
What is the cost of the machine?
60 lakhs.
Sir, there are different models. There are different models depending on what you're looking at. So it should be somewhere close to 70 lakhs, I would say 70, 75 lakhs. It's a workflow. We are talking of 4 different instruments with consumables, everything as a package.
So droplet has oil on the surface.
Correct, sir.
But when you heat it, don't they get bigger droplet?
No. So this is, I mean, we talk it as an oil, but it's a proprietary product. It has a lot of things added to it which maintain the integrity. Right?
Yeah.
So there are in fact a lot of publications also where we have shown how the integrity is maintained and how each compartment of a 20,000 droplets is same, is uniform, is of a same volume.
Thank you.
Yeah, so I have one, please sir. Do you have also any kind of provision for reagent printer?
We can talk about it, sir. We can talk about it.
Because we are thinking about in our hospital.
Yes, sir. So I am here, my colleague is here, sir, Ms. Akansha. We have a stall. If we can find some time after the talk, we can have a detailed discussion.
And also, what are the different hospitals where you have put this machine is used?
I think that he is available and people can discuss with him in the lobby or during the lunch. So I think with this, the session is over. Thank you speakers who are here, and thank you everybody for joining this session. Thank you very much.